Okay, so good afternoon, everybody. A very warm welcome to the fourth webinar in our series, where we're exploring how the engineering biology mission hubs are harnessing engineering biology to address real-world challenges. So my name's Pearson. I'm a senior portfolio manager for engineering biology at BBSRC, and in a few moments I'll be handing over to the Elemental Hub team to take you through the exciting work taking place across their consortium. But just before I do that, I want to spend a couple of minutes setting the scene in terms of why we consider engineering biology to be so important at UKRI, and also what we're hoping to achieve through these specific Mission Hub investments. Kicking off with a bit of housekeeping. So as this is a webinar, you'll note that your microphone's muted and your camera is turned off. We've also disabled the chat function just to ensure that all your questions are captured in the correct place. And in terms of questions, so you're encouraged to submit these to the Hub team throughout the webinar, and you can do this through the Q&A function. And then all the submitted questions will be collated and posted to the panel at the end of the webinar. And finally, we are recording today, and we plan to make the recording available on UKRI's YouTube channel sometime over the next few weeks. So please do keep an eye out for that. So engineering biology is one of our strategic priority areas for investment at UKRI. And the reason for this is because we strongly believe that engineering biology has potential to deliver huge benefits for the UK in terms of addressing major societal challenges, driving economic growth, and also increasing national security, resilience and preparedness. So as an emerging technology, engineering biology has potential to transform a broad range of diverse sectors, spanning agriculture and food systems, manufacturing and clean growth, in environmental monitoring and remediation, and also in biomedicine and in human health. And it's because of this broad range of applications that engineering biology is a technology that's recognised globally to be of major importance, with some reports predicting that it could contribute alongside other biotechnologies up to $4 trillion to the global economy over the next 10 years or so. So as part of our strategy to support the UK in realising engineering biology potential, last year, UKRI invested over £100 million in 6 engineering biology mission hubs and in 22 engineering biology mission awards. These investments were made through UKRI's National Engineering Biology Programme, with co-funding from the wider Technology Missions Fund programme, which has provided a substantial amount of investment to grow UK capabilities in critical technologies. The Mission Hubs and Mission Awards build on the UK's world-class engineering biology capabilities to really drive engineering biology to that next level of technology maturity, unlocking its potential across a broad range of application areas and ultimately delivering tangible impacts that make a real difference to how this technology benefits UK society. And now I'm very pleased to introduce one of the Mission Hub co-project leads, Professor Liz Rylott from the University of York, who along with her colleagues joining us today is going to explain how the Elemental Hub is harnessing engineering biology to address the growing need for critical minerals and metals in clean energy technologies.
Okay.
So thank you very much.
Thank you, Tom. Hello, and a lovely warm welcome to our Elemental Mission Hub webinar. So as Tom said, I'm Liz Rylott from the University of York, and I'll be giving the introduction before passing you over to my Hub colleagues. So on our title slide, you can see a lot of our lovely, friendly faces from our year 1 meeting in Manchester last year. We're a very friendly, approachable bunch. And on the right, you can see the geographical distribution of our Hub members from Durham, Manchester, UEA and Quadram, to the Natural History Museum, UCL, and the Universities of Surrey and Kent, nearer the bottom. So next slide, please. Before I get stuck in, here's an overview of the webinar sandwich fillings we'll be working our way through over the next 40 minutes. After my introduction, Joanne Santini, Jon Lloyd and Martin Warren will give spotlight presentations on some of the now maturing engineering biology-based projects that we've got coming through Elemental. Then after the spotlights, Anna Santos will explain how the Hub is supporting our early career researchers. And then Peter Chivers will explain how we're interacting with the wider community, that's you out there, and hopefully how we can work with you. So next slide, please. So in case you were unaware, we are not alone. The BBSRC has funded 6 engineering biology focused hubs, and here's a good place to give a shout out to our fellow hubs. We've got the IBEC, led by Fred Cullen at Cranfield University, The Microbial Foods Hub, led by Rodrigo Amaro at Imperial College. We have the Advanced Therapeutics, led by the wonderful Susan Rosser up at the University of Edinburgh, and the Glycocell, led by John Heap at the University of Nottingham. And finally, the P3EB Hub, led by Andrew Pickford at the University of Portsmouth. So to the other hubs, we have plans. We're looking to collaborate more deeply in the near future. So watch this space. Next slide, please. So what is our mission? What we want to do here is we want to harvest and develop living systems, plants and microbes to process environmental and waste metals. And we're doing this by enhancing the approaches for improving the accumulation of metals in those organisms, designing efficient and effective methods to extract, recover and recycle those metals, and exploiting both the novel genetic resources and components for industrial biotechnology to reduce metal contamination. And alongside this, a very much integral part of this is developing policy recommendations and guidelines for the use of engineered microorganisms in metal remediation. So this slide shows the circular economy of metals that our hub is working towards. And you can see inputting at the top left examples of our metal-rich waste streams, such as, they're not all here, but we have mining, urban e-waste and legacy nuclear. And many of these waste streams contain technology critical elements, including platinum group metals, nickel, gold, and rare earth elements. And rare earth elements particularly is a recurring topic in the BBC News, for example. And there are concerns that China's near monopoly on mining and refining these crucial materials is increasingly affecting global trade and technology. What we want to do is use sustainable approaches to develop security in our own supply chains. And you can see the points at which we're focusing and how they feed into the outer ring of processing, manufacture, use, and scrap metal recycling. And so on the next slide, you can see how these translate into our research themes. Next slide. First up is our bioleaching theme, led by the fabulous Dr. Anna Santos, and focusing on microbial metal bioextraction from mine and electronic wastes. Then the next three, we have biorecovery, bioprocessing, and biosensor themes. These will be covered in our upcoming spotlight talks, with Joanne Santini speaking next. I'm leading the only plant-based component of the hub here at York, and we're developing biomass crops to remediate environmental pollution and recover those technology critical metals and elements. Finally, Julia Stegman is leading the circular economy and responsible research and innovation aspects. And we consider RRI to be a crucial component to our hub and believe it's integral to fostering the trust and public confidence in science and technology that we need. And we have a lovely video from Joy Zhang, the professor of sociology at the University of Kent, coming up a little bit later. So our backstory, where did we come from? How did we get here? Elemental was born out of the BBSRC funded networks in industrial biotechnology and bioenergy. Perhaps you've heard them called NIBs. The Phase 1 Metals in Biology Network was one of 13 NIBs established in 2014 and led by our elemental big daddy, Professor Nigel Robinson. You can see his smiley face on the slide there up at Durham University. And this phase focused on the bioremediation and biorecovery of valuable metals. And it brought together a number of academics, industry and other connections into a valuable network. And this network continued into Phase 2, Elements of Bioremediation, Biomanufacturing and Bioenergy, also called E3B. And this was a consortium of universities with Durham as the lead alongside the Universities of Kent and Manchester, and established in 2016. This is one of just 6 Phase 2 BBSRC NIBs, and it accelerated the work of metals in biology, again specifically within the industrial, biotech and bioenergy areas, aiming to enhance, for example, metalloenzyme activities for new product development and improve the sustainability of existing products. So E3B finished in 2024, but do not worry, this valuable network has not been lost. It's been absorbed by our elemental hub and we're building it further. So next slide. We've got lots of examples, and a few are listed here, of projects that brought together academics and industrial partners through the Metals in Biology and E3B funding. You can see platinum group metals, biometallic catalysts, and rare earth elements are up there in those projects. So next slide, please. Thank you. So some of these Metals in Biology and E3B generator projects went on to secure funding in their own right and also become BBSRC impact showcases. And these are now being nurtured through the Elemental Hub. So I've probably wibbled on long enough now. So next up are 3 spotlight examples of research in our hub, starting with my fabulous colleague, Joanne Santini at University College London. Thank you, Jo.
Thanks, Liz. At least I'm fabulous this time. All right. So as work package 4 lead, the work package is divided into 2, but I'm going to concentrate on one aspect of it, which is enzyme-based metal and metalloid biosensors. And our, I guess our spotlight or our case study here is a enzyme called arsenide oxidase that we developed as an arsenic sensor, and I'll tell you about that. Next slide, please. Okay, so let's start with arsenic. So this is our metal or metalloid of preference. So it's known as the king of poisons. It's ubiquitous in the environment, and it's normally found in an insoluble form associated with other elements or other metals and in minerals. And so generally, not always, but anthropogenic activity results in the release of arsenic as the 2 soluble forms, as you can see in the top there, arsenate and arsenite. And it's these forms that are toxic and present in environmental wastewater, but also drinking water across the world. And I'm going to concentrate on arsenite. So next slide, please. So arsenic, king of poisons. So over 140 million people in greater than 70 countries consume water with arsenic concentrations above the WHO maximum contaminant level, which is 10 parts per billion or 10 micrograms per litre. On the right, you can see a map of the world. In red, you can see the areas that have the highest arsenic concentration. So these are arsenic concentrations of milligrams per litre in their drinking water. And then the lighter the colours, the lower the concentrations or no arsenic in the drinking water. And in white, you've got where there's no data. So you see pockets around the globe, North America, South America, Africa. But probably the biggest problem is in the Bengal Delta, with Bangladesh having 1/3 of the population consuming arsenic in their water above the WHO MCL. And so WHO has actually deemed the Bangladesh poisoning as the worst mass poisoning of a population in history. And so obvious signs of arsenic poisoning are in the bottom 3 images you've got there, hyperkeratosis of the skin, so basically skin lesions, but people end up dying of cardiovascular disease, various cancers, and lots of other things. So it's a huge, huge problem. Next slide, please. So the solutions, of course. So remediation, so I'm not going to talk about this, but you could remediate, you could clean up this water. But in Bangladesh alone, the water comes from 10 million wells, so remediating in individual sites is extremely difficult. The other option, at least in Bangladesh, is alternative sources of drinking water. So in that original map, generally those people across the world drinking arsenic contaminated water is where are drinking water where they have their own wells. Okay, so we've concentrated on Bangladesh and the need for a simple, easy to use, sensitive, cheap in-field testing system. So the current methods that are widely used are spectroscopy, which is obviously not an in-field testing, and chemical test kits that almost require a chemistry degree, so not particularly easy. And so the form that we've concentrated on, which is what's present in Bangladesh drinking water, is arsenite. So next slide, please. So this is where I come to a beautiful organism. So this bacterium is called NT26, isolated from a mine in Northern Territory in Australia. It's can use arsenite as a food source. So it oxidizes arsenite to arsenate. And with oxygen as it's an electron acceptor, so it breathes oxygen, uses arsenite as an electron donor, and only needs CO2 as a sole carbon source. And it's got 2 long flagella, and so its nickname is Mr. Tickle. So this is kind of the name that everyone calls this bug. It's actually got a formal name as well. Okay, so next slide, please. So it's taken a long time from the isolation of that organism back in 1999 to development of this sensor. And this is the arsenide oxidase. So this is the enzyme that converts that arsenide to arsenate, and where the organism can get energy from metabolizing this arsenic. And at the top, you see, I think, a beautiful structure based on X-ray crystallography, where you've got two large subunits in salmon and purple, lilac, and then you see the two small subunits at the top in grey and yellow. And this enzyme is a really complex enzyme. It's got lots of different metal cofactors in it as well. And so if you look at the bottom path, it shows you that when arsenite is oxidized to arsenate at that active site, there's molybdenum at that active site, and that molybdenum gets reduced, and then electrons are transferred from that molybdenum atom to a 3 iron 4 sulfur cluster, so a metal cofactor in the large subunit again, and then onto a risky 2 iron 2 sulfur cluster in the small subunit. And then what happens in the organism is the electrons are then transferred to an electron acceptor. So this is a protein called a heme C containing protein, cytochrome C, which also humans have in their mitochondria. And so this is then involved in generating energy in the organism. But we can use these transfer of electrons. Yes, next slide, please. Not yes slide, please. Next slide, please. We can use this to be able to measure those electrons, which gives us a direct indication of how much arsenite is in that drinking water. So what we did, we had to engineer the enzyme because we needed to produce a lot of it. And now organism of production is E. coli. So the enzyme was engineered, produced in E. coli. And then if you deposit the enzyme on a surface of an electrode, so what we have is very cheap disposable test strips. So we want these strips to be very cheap, that you can just throw away. The enzyme's deposited on the surface of that electrode. Then you put a drop of water on that test strip. You stick it into the little purpley machine on the right, very much like glucose sensors. So for people that use glucose sensors. So you put the water on the test strip, the strip in the machine, and then you measure the current. And the current that you get, because you're measuring electron transfer, is proportional to the amount of arsenite in the drinking water. And I think next slide, please. We're just going to end on a little movie that summarizes the whole story. thanks. Over to you, Jon.
Great, thank you, Jo. Thanks for that Spotlight 1 introduction. We're going to move on now to a slightly different area where microbes can be useful, not for sensing and detecting metals, but for recovering them from waste streams. So if I can move on to the next slide, please, Sophie. So much of the work that I'm going to talk about here is based on research that we've been doing in Manchester for a couple of decades now. So we're particularly interested in microbes that can metabolize metals, but in this case, we're looking at organisms that can respire metals. They live in the subsurface underground, no oxygen. They need another electron acceptor to support their growth, so they use metals, typically iron, but also a range of other metals, including critical elements that have become very interesting over the last few years. So we've been interested in the microbial ecology and physiology of these organisms, their environmental impact. But in the case of the Spotlight 2 area, understanding how we can take those natural systems, move them up from a natural environment, grow the organisms to scale, and then challenge them with waste liquors, and then use the natural processes to recover the metals. And through elemental, we're engineering those strains to make them more efficient and work better under more challenging process conditions. So the next slide, please. And this, in a nutshell, really encapsulates the processes that we're looking at. So we've grown an organism isolated from the subsurface. There's a couple of organisms we work with, one called Geobacter, one called Shewanella, but there are other organisms as well that we can look at. Typically, they'll take an electron donor for their metabolism, hydrogen or an organic acid. And then if you provide them with a metal, they'll reduce it. So on the top right, you can see what happens when you provide them with a fine grain rust material, which can cause sludge issues in a range of industries. Now, these organisms will reduce that sludge material, the Fe3 bearing mineral, into a fine grain magnetic mineral that can be removed very easily and then has higher value and a range of applications that we'll discuss in a moment. If you take away the iron on the top left and you look at something like a platinum group metal or a precious metal, these organisms will reduce those as well, and they'll take solution ions in solution, for example, palladium or gold in this example, reduce them in and around the cell, form nanoparticulate precipitates, which are retained within the biomass or around the biomass, and then you can use for other applications. These are particularly good for catalytic applications. Next slide, please. So some examples, and we'll skip on to the next slide, Sophie, thank you. Now, if we look at the iron, this is work we've done for a long time. This is quite an old paper, but it illustrates the sort of things that could be done. If you start off with a ferrihydrite sludge type solution, this rusty material, add your microbes with a cheap electron donor, maybe a few other simple additions. Within a few hours, you'll start to produce the magnetite nanoparticles that you want. And in this example, we've titrated in small amounts of cobalt to give us a designer magnet, which is in that example was good for or tailored for medical applications. We've skipped another slide, but I'll just cover what was in that one very quickly. Oh, okay, that's fine. We're on this one now. Obviously, we know these systems are scalable. This was work with the Centre for Process Innovation, where we're able to go from half-litre volumes up to 5 and 50 litres in their industrial plants. So we know these systems are eminently scalable. Next slide, please, Sophie. Thank you. And then I can also just illustrate some of the other applications that we've looked at for these materials. They're fine grain, they're magnetic, you can recover them. They're also catalytically active and very good at treating contaminants. For example, toxic metals, radionuclides, organics on those studies. And the next slide covers some of the other catalysis reactions, for example, upgrading low-grade fuels, synthesis of materials, and more recently, aligned with the elemental work, titrating in rare earths into the system to give you materials that could be used in electrochemical cells for clean energy applications. Okay, next slide, please. Now, what we've found out over the last few years with colleagues also in elemental, Juma on the author list at the bottom left is based in Surrey and helps us with life cycle assessments for our processes. It becomes very obvious you need to use waste materials. And that's something that's embedded across all our projects in this area in elemental. Next slide, please. Now, we know certainly with iron we can use a lot of waste materials from industry and contaminated environments. So, for example, this study showed that you could use acid mine drainage waste materials, and that work's now ongoing with a parallel project to elemental, getting these systems to hopefully work on materials that are available at scale with help from the Mining Remediation Authority. Next slide, please. Okay, so for the last few slides, I'd like to sort of shift focus and talk about the higher value metals that we're working on now. Next slide, please. So that's taking us into the critical metal contaminated wastewater area. And this allows us to look at metals that are required for the clean technology applications, the batteries, fuel cells, motors, wind turbines, photovoltaics, etc. Your rare earths, your transition metals, including cobalt, nickel, and your PGMs. And these are significant concerns for various reasons that Liz has mentioned earlier on. Next slide, please. And tackling these metals also helps us tap into an emerging problem in the UK and also overseas, and that's the e-waste mountain that's growing. We're particularly good at producing e-waste, 24 kilograms per person per year from these estimates that were linked into a House of Commons committee report quite recently. And they contain a lot of metals that you do not want to throw away, particularly critical metals. Next slide, please. So the one we've worked on most over the years is palladium. This goes back more than 20 years. So if we take the iron out, put PD2 in, you'll get palladium in and around the biomass. This is using one of our metal reducers. Next slide, please, Sophie. And that material can then be used for a broad range of catalytic reactions for industry. Next slide, please. You don't normally get a metal on its own, so you're not just going to get palladium or another PGM, you're going to get other metals as well. And we know that if you've also got mixed metal streams, you can make some very interesting materials. So with palladium and gold or silver, you can make some very interesting materials for these coupling reactions that are shown in this paper here. So, the mixed metals can actually be a benefit in these process streams. Next slide, please. But there are challenges. The solutions can be quite difficult to what these organisms normally see, and that's where engineering biology comes in. So at this point, I'll just highlight very quickly a parallel award project that we're working on for 2 years with JM, which sits alongside the elemental work. So James Johnson Matthey, a company I'm sure many of you are familiar with, have a significant interest in the platinum group metals. Next slide, please. And in that study, we're using some of the engineering biology approaches that Martin will talk about next to get our cultures working properly in their industrial effluents, and we're testing the materials that we're collecting from JM's effluents for catalytic reactions, and throughout this doing LCA and environmental impacts to make sure that these systems are useful. Next slide, please. In elemental, we're also working on other metals, including copper, which can be reduced from copper 2+ down to copper 0. And you get these lovely nanoparticles around the cell. This is Schuanella. There's a copper 1 shell around that copper 0 nanoparticle, so you can do catalytic reactions. We're doing more work on this through elemental and also producing these materials for partners that are interested in these nanoparticles for other environmental applications. Next slide, please. Through elemental, we've been able to show that the hydrogenases are involved. They're driving this system. They're essentially the reductases for copper. And the next slide shows that this enzyme is also important for reducing gold. And that's allowed us to work with another commercial partner, in this case, Mint Innovation, a New Zealand-based company who are the first on the scene, amongst the first on the scene for bioprocessing of electronic waste. So there's a proof of concept study here to show that these cells can work on their effluents. And if we look at the next slide, you can see how that might fit into a process sheet that they would have. And other companies doing this sort of work would use a range of conventional technologies to start to leach out the metals, maybe bioleaching as well. And then these organisms can step in at the end for the final polishing and recovery steps. Next slide, please. So last couple of slides in this area just emphasize that these biomineralization reactions are important, not just for metal recovery, but also for bioremediation. And this links into Liz's work package. And in particular, we're very interested in helping in the nuclear area. We've got long-standing interests with Sellafield and other stakeholders where there's a £150 billion program of work that will last many decades to deal with our legacy contaminated soil sediments and waters. And the idea here is to use these same biomineralization reactions to immobilize radionuclides within contaminated sites so that they're stabilized and will not leave those sites. So the next slide, please, Sophie. And just to emphasize here, what we're doing is running a series of projects that are ongoing through other activities where we're looking at a range of options, including biomineralization with our industrial partners. And now with Elemental, we can go in and we can try to tailor those pure cultures and communities so that we're more confident that they can survive under the harsh conditions you would find in a nuclear regulated site. So this work was very timely and allows us to slot into the nuclear remediation area seamlessly. Last slide from me, Sophie, please. And these are just some bullet points just to leave you within this area. So we're very interested in, through Elemental, in one of the work packages, in engineering and scale up of these biomineralization processes. We're interested in working on anything from iron through to the higher value metals and critical metals in particular. We will integrate these processes across the Elemental work packages, so we'll also be integrating into bioleaching, scale up, sensing and life cycle assessment work. And the idea here is to help support UK remediation targets and also the energy transition in a circular economy for these critical metals. So a bit of a run through a range of projects. I hope there was something there that was of interest. Please contact us if that's something that you'd like to build on. And I'll pass on now to Martin for Spotlight 3.
Okay, thanks very much, John. And I'm going to take you through some of the work that we've been looking at with regard to how we can improve the efficiency of metal uptake and metal processing in fermentation systems. And this is both applies to both eukaryote and eukaryotic systems. But a lot of the genetic systems that we utilize to understand what's happening with metal uptake and metal import and export actually apply to a whole range of our biorecovery and solubilization processes. So you'll see examples of that genetic approach as I go through. But next slide, please. So the reason why we are in the position we're in is because we've now got a much greater understanding of how and why metals get incorporated into proteins. We understand more about the free energy of binding of metals within cells and how that leads to the regulation of the metal importers and the metal exporters. And what we know is that around 50% of all enzymes require specific metals for activity. So in order, if you're overproducing a protein that requires a particular metal, you've got to understand whether that metal is, or whether that protein or enzyme is going to be incorporated with the correct metal. And by understanding those processes that take place, what we want to do is to ensure that you do get the correct metallation of an enzyme or a protein. Or if you're taking a metal into the system, if, for instance, a biorecovery, can we store it in a particular area within the cell so that it is out of harm's way? And key to that, of course, is understanding how the metal is taken up, how it's handled, how the oxidation state is controlled, and how that then leads to that incorporation into that particular protein or process. Industry is really interested in this because obviously, if you're doing large-scale fermentations, if you have excess metal left in the end of your fermentation broth, in essence, you've got to undertake quite an expensive process of removing that metal before you can lose that waste through waste water management systems. So being able to be efficient with the metal uptake and its incorporation is important. And if you can enhance that so you get nearly all of the metal incorporated into the right place at the right time, obviously is going to be very important. And that's what a lot of our work is focused on. If we go for the next slide. So the way to do that is, especially if you're dealing with a cell that doesn't normally handle a particular metal, is to ensure that you have the correct metal importer and the correct metal exporter. And then within the cell, you've got to have a way of handling that metal. Now, it can just be a protein. It could be a bacterial microcompartment, for instance, that's going to aggregate the protein, or a ferritin type molecule. So it's understanding that relationship between the import and the export to ensure that you get the correct homeostasis of the metal within the cytosol, because what you don't want to do is to accumulate the metal to a toxic level, which kills the cell off. Next slide, please. And the way that we do it, and the next slide, the way we do that is to use TRADIS, which is a transposon-directed insertion sequencing approach, where you can take your cells, and in our case, when we're dealing with metal, where we're trying to look at the uptake of a metal, for instance, we can expose a transposon library to increasing concentrations of the metal, and then sequence the surviving strains to identify what mutations have taken place to ensure that adaptation to those higher metal concentrations. And that way gives us not just the identification of a single protein, but of all the proteins that have been affected during that adaptation process. So if we go to the next slide, we've used this approach, for instance, in our understanding of improving vitamin B12 production in E. coli. Now, E. coli is an organism that doesn't normally make vitamin B12. Vitamin B12 has a cobalt ion at the center of the molecule. E. coli doesn't normally utilize cobalt, so it doesn't either make vitamin B12 and it doesn't handle that metal. And what we wanted to do was to adapt the strain so that it would make vitamin B12 and would take and handle cobalt very efficiently. Next slide, please. So we know that E. coli can take up cobalt, but the mechanism isn't clear. We know that cobalt is actually quite toxic to the cell. So once you get above 150 micromolar concentrations, it starts to interfere with iron metabolism. So what we wanted to do is to find ways that we could optimize B12 production in the presence of exogenous cobalt. Now, our advantage is that we were able to take all of the B12 biosynthetic genes and put those into an E. coli strain. And then what we found is that as we added cobalt to that strain, we were finding that cobalt was going into the E. coli. And the E. coli were producing B12, but we had to increase the level of B12 to start to get that and make that process more efficient. And the issue is that actually the more B12 that we wanted to produce, we were getting to those very toxic levels of vitamin B, of cobalt for optimal B12 production. So next slide, please. And the slide after as well. So our way to do that was to actually take our E. coli, our vitamin B12 E. coli producing strain, and we subject it to a slightly advanced TRADIS system. Now, the TRADIS system has been developed at the Quadram Institute by Mark Webber and Ian Charles, who are both part of our engineering biology hub. And what they're able to do is make that very efficient library and then challenge our B12 producing strain with increasing concentrations of cobalt, do all of the sequencing and identify the genes involved. And what they found was, if we go to the next slide, is that, you know, again, you're not identifying one or two genes, you're actually identifying probably around 100 genes that are being affected. And we, through that process, we were able to identify the endogenous cobalt transporters, both the importers and the exporters, where the cobalt effectively was piggybacking, for instance, on nickel transporters and magnesium transporters. We also identified all of the genes that were affected to do with iron and sulfur metabolism and iron sulfur centers. It also had an effect on fatty acid biosynthesis and iron siderophore production. It also stimulated stress responses, and it also affected iron regulation. And we've been able to take all of that knowledge and start to adapt our strain so that it suddenly becomes optimized for handling cobalt. So if we go to the next slide, what we've been able to do is to look at our previous strain where we were struggling to get B12 production at around a concentration where we're starting to be starting to be toxic. And by introducing specific cobalt transporters and modulating our export system, if we go to the next slide, we see that we suddenly drop our cobalt, our B12 production range from 160 micromolar of exogenous cobalt to 20 micromolar exogenous cobalt. We're modifying this slightly so it's not quite so sensitive. But what this tells us is that we now have a way of producing B12 using a very limited and minimal amount of cobalt, such that all of the cobalt that's absorbed by the bacterium goes into vitamin B12. So that's just an example of one of the processes that we're playing with. But we want to take that same kind of approach and that technology as we try to incorporate and recover a lot of these rare earth elements. Okay, that's enough for me, and I'm going to pass you on to Anna with the next slide.
Thank you very much, Martin. Hello, everyone. And I'm here to talk to you about Elemental's career development portfolio. And this portfolio was developed based on 3 pillars: capability, culture, and connectivity. And that was, and the aim was to empower those working in Elemental to acquire new skills, develop existing skills, knowledge, and how to effectively transfer the knowledge so they can excel in their roles, but also to develop an inclusive and supportive research environment. Next slide, please, Sophie. So within the capability, to develop the capability, we have 2 main aspects: the technical training via workshops and seminars that are conducted by industry experts and also experienced researchers, to enhance specific technical skills such as data analysis, programming, the use of online platforms. And all of these are tailored to those working in Elemental and what the project needs are. The other aspect is the hands-on training sessions in laboratories and in pilot plants. So those working in Elemental have the experience and the opportunity to familiarize themselves with this type of research environment, but also being exposed to new pieces of equipment, tools, and techniques. The next pillar, next slide, please, Sophie, is the connectivity. And to develop connectivity, we are promoting interdisciplinary training, and that is to facilitate the interactions between the Elemental members from different disciplines, different backgrounds, and to encourage the exchange of ideas and to promote that collaborative work. And also in terms of communications training, and that is to ensure high standards in all events that we organize and that we participate, including both academic conferences and any public engagement that we have or planned to run in the next few years. Next slide, please. To develop a research culture, we are looking at professional development in the form of mentorship programs, and that is for the experienced researchers and industry experts to really help the younger generation, guiding them through their research, but also sharing them experiences with the hurdles of project management. As we all know, it's not always very straightforward, so that it also brings an element of professional development in there. We also work as a team, and our activities that we promote within Elemental, it promotes the interaction not only between the co-Is and the early career researchers, but the ECRs themselves as a cohort. Ethical and responsible research training, we are going to hear from Joy later on through a video, but that is to support a more inclusive and effective stakeholder engagement as well. Next slide, please, Sophie. We also have other opportunities under the Elemental's portfolio, and these are a sum of money for enterprise business interaction vouchers and summer research projects, and all of these are there to promote new collaborations and interactions, not only within the hub, but also with industry. And we are looking to work in partnership with other mission hubs and awards to help deliver a broader training program that hopefully will cover all the aspects of engineering biology. Next slide, please, Sophie. And Elemental is also looking into the future, and we need more scientists that are as passionate as we are about our job. And Elemental is seeding the next generation of researchers. And this year, we funded 3 summer research projects, and these are the students already in place working in each of the institutions. There will be more opportunities like this available in the future years, so watch this space. And the amazing thing about this summer projects in particular is that these students that have the opportunity to be in a research environment and learning new skills, but also it gives our research early career researchers the opportunity to gain leadership experience, but also to supervise. So that is a win-win for both sides. Next slide, please, Sophie. And speaking of our early career researchers, this is our fantastic team of ECRs representing each of the institutions part of Elemental. And I'm sure all of them would be incredibly happy to talk about that research topic if that's of interest. And with that, I pass it to Peter.
Thank you, Anna. So to close out in the last few minutes, I'm going to talk a little bit about how Elemental is looking outwards. So here are 2 examples looking both within the UK and more broadly. So shown on the left is a picture of Dame Angela McLean, the Chief Scientific Advisor for the UK government, visiting with Liz in York as part of a recent tour she did around the UK, learning about what's going on with engineering biology, and who's published a report recently advocating for engineering biology research. And on the right is a picture of a joint workshop we had with members of the Critical Minerals Institute based in the US. And this was a 2-day workshop hosted and co-organized by Anna at the Natural History Museum, where we covered a range of critical minerals issues, including rare earth elements, and there are various outcomes to this that including additional funding for future meetings, drafting of a position paper. So we hope to continue these interactions as well as build interactions with similar groups in other countries. Next slide, please. As Anna mentioned, we have an important RRI component. And this is to enable us to communicate more broadly with the public about the roles of engineering biology and to reach a consensus of how it can be employed in society. So Joy unfortunately can't be with us today. She is on another continent attending a conference, but she's recorded a short video to describe her work within Elemental.
Hi, I'm Joy Zhang. I'm the director of the Center for Global Science and Epistemic Justice at the University of Kent. I'm also the responsible research and innovation lead for Elemental. So in addition to support more traditional RI reflection within the project team, we're also acutely aware that translating scientific advancement into real-world benefits require the support and collaboration across all sectors of society. Public understanding of and attitude towards application of engineering biology are critical, not only for translating Elemental's finding into practice, but also for providing valuable feedback for the broader field. And such feedback can help with research and industry design, as well as inspiring more thoughtful research questions going forward. This is why we're aligning Elemental with a new odyssey of public dialogue we're promoting here at Kent. And this new odyssey is an acronym for an open, deliberative, enabling, sensible and sensitive, and innovative approach, which is designed to support more inclusive, responsive, and diplomatically agile forms of public engagement. This initiative is particularly relevant to Elemental, which addresses both the geopolitical and increasing cultural concern over the sustainable use of critical minerals. We aim to engage stakeholders across the mineral sector, spanning research, policy, civil society, and relevant communities in this effort. We also believe that this odyssey will positively impact all 6 BBSRC engineering biology mission hubs because it promotes a new cultural and practice shift that does not treat ethics as dogma, but as a living document co-authored by members of our contemporary communities, and is a living document that reflects both the opportunity and obligations of our collective potential. Thus, we welcome all engineering biology mission hubs, as well as anyone who's interested in the application of science, to join us in this new odyssey.
So as you can imagine, we're very excited to have Joy as part of our team, and please look forward to more information about Joy's activities coming in the future from Elemental Communications. So we are also keen to develop cross-hub interactions. There's overlap between all of engineering biology and some hubs in particular. So first of all, we are supporting a number of PDRAs who are members of the broader Elemental Hub to attend the EBIC ECR conference, which is taking place next week in Edinburgh. And this will be an excellent opportunity for career development there. We have interactions with PE3EB, where we are co-applicants with a co-I on PE3EB for this EPSRC funding call. And additionally, we have provided support for some BBSRC engineering biology focal award applications for PhD training. And we're looking to build more of these in the future, although it's not yet appropriate to mention what some of those are. Next slide, please. So we also have a number of industrial partners, past and present. These are obviously critical to the success of engineering biology, because these are the agencies that have waste streams that can be of interest and need remediation recovery, as John has alluded to. And so finally, here is where, next slide please, where some of you can come in is by contributing to our efforts to build these interactions between industry and academia to generate new projects, developing technology, scaling up. And so this is through our first round of business interaction vouchers. This call has just opened. And so we're looking for projects linked to engineering, biology, and metals recovery. Obviously, particularly interested in those sorts of metals for which there are geopolitical considerations, as Liz mentioned early on, for example, rare earth elements. So you can find more detail about this on our website, but just to highlight here that the deadline for these applications is the 30th of October. So having dangled that carrot in front of everyone, I'd like to bring a close to the presentation, and we would be happy to take any questions.
Okay, thanks very much to all of the Elemental Hub team members here today. So if I could just ask you, Sophie, to stop sharing your screen for a moment. And we will move into the Q&A section of the webinar. So if you did have any questions for the team, please do submit those through the Q&A feature. You should be able to see that at the top or the side of your screen. And we do have a couple of questions submitted. So the first one is regarding the B12 production using cobalt. So if I understand correctly, the intention is to clean up cobalt contamination. But is the B12 which is produced by the E. coli meant for commercialization? And if so, is this competitive to how B12 is usually commercialized?
Well, I'll take this one on the nose. So yes, I mean, B12 production is actually has similar geopolitical issues in terms of its production as the rare earth elements. 90% of it is controlled by China, and there's only one factory that produces B12 outside of China, in fact. It's a remarkably expensive nutrient. Our strains are competitive with commercial strains, and it's one of the aims and objectives is to try and find a way to produce British B12, or at least UK-based B12, which we think we can do in an affordable manner. So the key thing from our point of view, and the advantage we have over the other commercial producers, is the fact that we have systems that are going to be environmentally very friendly. We won't have any metal in the waste stream, and we've also developed improved methods for B12 extraction and purification, so we can do it in a very rapid and timely manner.
Okay. Thanks very much, Martin. So we have another question here, and this is a general question to all speakers. So how scalable are microbe-based bioremediation or extraction of rare earth minerals? Can they compete with national and/or global markets so that these potentially clean technologies can drive economic growth in order to sustain themselves?
Okay, thanks, Tom. I'm going to throw this across to Jon on the microbe front, if that's okay with you. You're muted, Jon.
You're on mute, Jon.
How's that? Can you hear me now?
Right.
Apologies for that. Yeah, no, I'm happy to have a first go at that. Well, you know, potentially very scalable. I mean, we know, you know, in the bioremediation area, and this is probably a wider point than the rare earths, but, you know, certainly the bioremediation sectors that do use bio approaches and have done for many years. So, you know, they can be used at scale for cleanup. For recovery and processing, which I think is perhaps more pertinent to the rare earth question. Again, you know, potentially eminently scalable, but these are early days with these systems, to be honest. But if you look in the bioprocessing area, you know, there's things like, as Anna will tell you, in the bioleaching area, you know, these have been used at large scale for many, many years. So, you know, there's no reason why potentially they couldn't be used for other metals and other processes. I guess one of the particular advantages around rare earths is that you might be able to solve the problem of selectivity, and there's a lot of interest in that area. So if you can separate heavy from light elements, then you know, that could be very useful, and certainly the biological systems do seem to have that potential. So I wouldn't want to overpromise. I mean, I think it's early days for rare earths, but there's a lot of interesting work that's being done and an awful lot of potential. And if you can compare it to other processes and sectors, there's no reason why if they don't work at the smaller scale, they couldn't be upscaled.
Okay, thank you very much. So I can't see any other questions in the Q&A at the moment, but seeing as we are nearly at time, perhaps I could ask one final question. So if anybody wanted to get in touch with the Hub to find out more or to ask any other questions, what's the best way to do that?
Okay, well, we had a 3D barcode there. We have our Elemental website as well, and all our emails, our contacts are on there too. Has anyone got the address of the website to hand? elemental.co.uk, I'm pretty sure it's something like that. And of course, they can get to you through the UKRI BBSRC webpage as well, where we have contacts listed on that. And then, Tom, I believe that this webinar is going to be posted in a couple of weeks somewhere too.
Yeah, that's absolutely right. So within the next few weeks, it should be available on YouTube. So hello to anybody watching back on YouTube.
Okay, thanks, Tom.
No worries. Okay, well, we are at time now. So thanks very much to the Elemental Hub team for presenting today. Thanks to all the attendees for coming along. We are taking a break now in the series over the summer, but we will be back with the next webinar in September, which is going to feature the Glycocell Mission Hub at Nottingham, led by Professor John Heap. So thanks very much, everybody. Have a great summer. Cheers.
Thank you.
Thank you.